Morning. My name is Krista Riley. I'm with Spokane County, and I'm here to introduce our presenters today. The talk today is on non-vegetated bioretention soil media study. Spokane County is actually a partner and in this project and working with Evergreen on this, so it's an honor to present this project today. I'm just going to give you a little bit of background about our presenters. So Aimee Navickis-Brasch on the left here is the president and principal engineer at Evergreen Storm H2O with over 30 years stormwater management experience, research, design, planning, permit compliance support, and training. She holds a bachelor's degree in mechanical engineering from Gonzaga University, a master's degree in civil engineering from Washington State University, and a PhD in civil engineering from the University of Idaho. Aimee is serving as project manager or principal investigator for the bioretention study. And then our second presenter is Mikala Lindgren. Mikala is a stormwater engineering technician at Evergreen Storm H2O with over 2 years stormwater experience, focused on a variety of field equipment to collect stormwater system asset data, as well as water quality and quantity data. She holds an AAS water science degree from Spokane Community College, along with certificates of GIS, water and wastewater sampling, and BMP inspection and maintenance. Mikala is a field lead for the non-vegetated bioretention study, responsible for sample collection as well as equipment maintenance and calibration. So we'll have 10 minutes at the end, so please hold your questions to the end, and then so that everyone online can hear when I run the microphone to you guys so that it's all consistent. So please welcome Aimee and Mikala.
Well, thank you for attending the presentation. I'm going to give you a quick overview of our presentation first. First, I'm going to start off with going over the study team and who's contributed and been involved in the project. We'll go over the goals of the project and a little bit of background information on it, provide you an overview of the project timeline as well, get into the study design, including a review of construction, because construction happened this fall. We've got some great pictures and experiences from that. And then I'll provide a review of the data we've collected so far and the results we're currently seeing. And then we'll provide a summary of next steps and some lessons learned that we've developed on the project. The pictures that are shown on the right, this has been a bioretention research test site since the fall of 2013, and that's what it looked like when it started in the upper left corner, and it was completely rebuilt into a vegetated site. We did a media thickness study and did a side-by-side comparison, and then rebuilt again in the fall of 22, and is currently non-vegetated. And we'll tell you a little bit more about the current design for that in this presentation. So the study team, the lead entity for the project is the City of Spokane. They were also the contractor. Their operation and maintenance team came out to the site and built the test site for us. Evergreen installed all the monitoring equipment, but they dug the hole, put in the media, did some of the big construction work there. The partners on the project are Spokane County and the City of Spokane Valley. Evergreen Stormwater is the prime consultant for the project, and Curtis Hinman and Associates is also a sub-consultant working on the project. This project was funded with an Ecology water quality grant as well. And we also have a university partnership. Since we're on Gonzaga's campus, we have had 35 senior students work on the project as part of their senior design project. And I think there's at least 2 in the room, one up here. And I think I saw Bella back there, the 2 of the 35 sitting in this room who, as part of their senior design project, worked on some aspect of it, helped collect data, and then also just advance the knowledge of bioretention research in Eastern Washington. So thank you on that. We have a very small technical advisory committee composed of the lead and partner entities, Ecology, our sub-consultant in King County, that help provide feedback on the deliverables for the project, as well as thoughts and ideas on different items that have come up on the project. Folks from Ecology that are managing the grant as well. And I need to also acknowledge the critter that we cohabitates the site with us, the marmots, who we have a love-hate relationship with. Mostly we exist in harmony, but occasionally we've had trouble with them climbing in through our equipment and hibernating around a pressure transducer and then getting to big when they climbed back out and having to destroy our weir in the process. And we knew it was them because they left gnaw marks in the weir that they ripped out. So anyway, so they coexist at our site. We've, over the 12 years we've been doing this, we've learned pretty much have harmony. If you talk to them, they may tell you a different story, but we seem to be doing pretty good now.
So our project goal for this project was to evaluate whether the non-vegetative bioretention meets the TAPE treatments and performance goals. The big project driver for this project was for locations with hot and dry summers. Obviously, a non-vegetated cell is desirable. This is a lack of need for irrigation, as well as not keeping the plants lively. We're comparing two different types of media, a high performance biosoil media that is made up of an 18-inch primary layer and a 12-inch polishing layer, and then a 60-40 biosoil media that is an 18-inch mixture of 60% sand and 40% compost.
Is everybody in here familiar with tape? Is anybody not familiar with tape? Okay, so we did have this slide just in case if you're sitting here, I don't know what you're talking about with tape. So just real quick, TAPE stands for Technology Assessment Protocol Ecology. And basically what it is it's a set of standards that says this is how you will evaluate the effectiveness of a BMP. And it tells you exactly what you need to do. It defines things like what is a qualifying storm? And that includes the duration of the rainfall event, the depth of rainfall, when the storm starts and when it ends. It defines those specific conditions. It tells you how many samples you have to collect. So we have to get 15 samples from qualifying rainfall events on this project. And it also defines the treatment performance criteria. So it says how much each bioretention media needs to reduce of specific pollutants. And so like for TSS, there's an 80% reduction. It provides that specific standard for what's working and meeting the permit requirements. Back to Mikala.
So our project began in November of 2023. This is when contracts were executed. And then our study preparation began in December of 2023 and kind of moved through that next year, December of 2024. We started construction this October and wrapped up construction in December, and then began began sampling here in January of 2025. So our data collection will continue through December of 2026, and then we'll begin our reporting phase. And then our project will be reported and projected to end around June of 2027. Here is our study design and test site location. So we're located on Gonzaga campus, as we mentioned earlier, in Spokane, Washington. Our runoff comes from a parking lot just outside of a soccer field. Our runoff basin is 0.61 acres. Something that's interesting about our test site here in Washington is we get 4 seasons. So we are figuring out the struggles of rain on snow events and what all 4 seasons hold with falling leaves and these blooms that are also happening here in the spring. Here are our first set of construction photos. This photo here on the left is of day 1. This was that previous test site that was ripped out, the big hole in the ground. And then we constructed our manhole as well as a berm that separates the 2 cells. They're installing a conduit pipe, which is where the sampling tubing runs through that's connected to a set of ISCOs. Then they're stacking the top of that manhole in that last picture there on the right. Here is a 30 milliliter HDP geothermal liner that they're laying over the entire test site, as well as the perforated pipe that they're laying in both of the cells. The City of Spokane actually created those perf pipes, so I got to watch them cut the individual perforations in the pipe, which was pretty incredible. The last picture is of them setting the media. The media came in these large bags, some with ties at the bottom that could be ripped open, and then some that needed to be cut open. You can see in this video them kind of releasing the media, and then the digger will shake, and the rest of the media will spread out evenly along the bottom, and then they used rakes to make sure that the elevation was accurate across the floor. This picture here on the bottom left is of both of the medias laid. So this was after they were raked to grade and made sure that they were level across. That high performance biosoil media on the left, and then the 60-40 biosoil media on the right. They were covered in a 3/4 inch basalt rock. This is requested by Gonzaga campus. It matched other rock that covered their gardens around campus. And then the lower picture on the right is of a completed test site. So we completed it with an edging circling the site and then made the rock go all the way to our monitoring monitoring vault. So that vault that I was talking about here in picture 1 houses all of our monitoring equipment. So this is our 3 ISCOs as well as our data logger. This is also where our pressure transducers are plugged into, and they have dehumidifiers, which are hanging up on the wall there. Picture 2 is of our volumetric weirs that are throughout the piping of the system. 3 is of our inlet sump. This is where our pressure transducer sits for the inlet that measures the flow. 4 is of the catch basin in the parking lot, where runoff comes directly off the parking lot into that catch basin before it enters the system. And then the last picture is of our manhole. So the well that you see in the foreground of the photo is the 60/40 media, and the well that is deeper into that manhole is the high performance biomedia. The tubes that you see kind of circling through the picture are those tubes pulling the samples into the ISCOs. And then in the well farther away from that is where that pressure transducer is sitting measuring flow. So as Aimee mentioned earlier, we're targeting 15 storms to sample. We measure in composite samples, so the ISCOs pull into one large jar, and then we split this jar into varying amounts for sampling, and then the samples are sent to 2 labs, one in Spokane, that is Anatech lab, and then we ship our 6 PPD samples to a lab, Manchester Environmental Lab in Port Orchard. Then here you see the required sampling parameters for tape as well as the screening parameters. We were afforded a 6 ppdq amendment, and that is where we are getting that 6 ppdq sampling, and we're only testing the screening parameters 3 times over this course.
Mikala. So I'm going to walk you through the results, starting with total suspended solids. So we've only had 3 qualifying events since we started in January that we have data back for that we're able to include. We do have 5 for 6 ppd. But for the 3 events we've had for total suspended solids, we've had a range of TSS coming in from about 130 down to 35 milligrams per liter, with our median being at 77 milligrams per liter. We've had a really good reduction. Tape, which is in your bottom right. The criteria for that is if it's over 100 milligrams per liter, our reduction needs to be 80%. And if it's under 100 milligrams per liter, we need to have an effluent concentration of 20 milligrams per liter or less. And for all of the 3 events that we've had, we've either hit the 80% or been below 20 if our influent was below 100 milligrams per liter. So we've had great results with TSS. I'm going to talk a little bit about pH and hardness. We live in 4 seasons, and then we have some different conditions with snowfall that occurs. So on February 22nd, we had a rain on snow event. It had snowed all month. Gonzaga is very diligent about applying their de-icer 6 times that month before between rainstorms and piling their snow up in the parking lot. And we didn't have a ton of snow, but we did have piles of snow that were all over the parking lot. So we had a great source of pollutants everywhere that had piled up. So we were all over the storm, very excited to get this rain on snow event. We got the data back, and the influent hardness concentration was 2,700 milligrams per liter. To provide context to how high that is, it's typically around 20 for other events when they're not applying de-icer 6 times. So we do have this extreme condition that can occur with all of this, and it's magnesium chloride that's coming into these ponds that we wouldn't normally be seeing for that. Most of the time with hardness, you actually see a slight increase as it goes through the media, especially the 60/40. For that event, we definitely still saw a decline. The other ones was a slight increase. pH is typically around 7 coming in and just a little above 7 coming out. For that particular event, we don't normally do hardness. That's a new tape requirement that we added in. But for the event where we had 2,700 milligrams of hardness coming in, our effluent pH from the 60/40 was actually just below 6. I was surprised to see that correlation that might be there between the 2. Typically, like I said, it's around 7. Total phosphorus reduction, for the HPBSM, we expected it to have a reduction. Polishing layer was developed just to target phosphorus. Tape requires a 50% reduction. We well exceeded that. Wasn't a surprise. 60-40, we weren't expecting a reduction. It's known to leach phosphorus, so it's not approved for phosphorus reduction. We saw that in storm 2 or 3. We did see a reduction in storm 1. That's pretty rare to see. And I suspect that's because we had a influent hardest concentration of 2,700 milligrams per liter, all that magnesium coming in that help bind and potentially form participants that are filtered out easier. But still didn't meet tape. We didn't expect it. For dissolved metals, looking at dissolved copper and dissolved zinc, tape criteria for dissolved copper is a 30% reduction. With the HPBSM, it exceeded well over the 30% and met it. 60-40, it did not. It was leaching copper. Not a surprise. That's been seen in other research that's been done too, that the 60-40 doesn't do a great job with copper. It definitely reduces the toxicity, and we know that from other studies. Just the concentration, we don't see that reduction. Dissolved zinc, tape criteria is a 60% reduction. For the HPBSM, they exceeded that, did fantastic with it. 60-40 didn't quite hit that. In Eastern Washington, we've had a couple bioretention studies, and we haven't quite hit the tape criteria for zinc. I'm understanding that's been a little bit better on the west side. I'm not quite sure why that is, but we're not quite hitting it there. For 6-PPD, we have a little more flexibility. We're just trying to get data. That's the goal right now. Let's get 6-PPD data, so we're not just focused on is this a qualifying event or not. So we've had some events that don't quite have enough volume to get all the samples, so we've just pulled 6-PPD. Our concentrations have ranged from about 250 down to about 40 nanograms per liter, with the median being at 163. We have a high-use parking lot. It's packed during the academic year, a lot of turnovers of vehicles during the day as well. During sporting events, it's packed as well. So it's a high-use parking lot that it's being used there. Both the HPBSM and the 60-40 did fantastic with reducing 6-PPDQ. We had over 98%, 97% for those. So that was really great data. And it's very consistent between all of the storms we've had that we've seen that reduction. Okay, a little bit about next steps. Next step is recommending maintenance procedures. That is in our scope. How do we handle these non-vegetated BMPs so we don't get sediment buildup, grass that grows on it, and pretty much it's a vegetated rocks, mess. So how do we deal with that? When it was installed in the fall of 24, immediately we had the leaf drop, and I'm like, oh, that's going to be such a pain. And then we got the spring and we had the seed drop, and I thought, oh, I'd rather have the leaf problem. So we'll be working on developing guidance for how you address this in this non-vegetated system, and then as well as trying to prevent plant growth, that sediment buildup. We'll be continuing sampling until December of 26. That's the duration for our project. As soon as we finish January of 27, project switches over. We'll have a new senior design team, and we're going to get to stress test the site with de-icer. I'm very excited. We're running 6 storms of stormwater through it, and then doing another 6 with de-icer and doing a comparison between the two to kind of understand are we getting differences in the data because of all that de-icer that's applied. The final reports will be done in June of 27 for both the tape study as well as the de-icer stress test that we'll do. A few lessons learned from the project that we've had. One is making sure your chokestone is really washed and free of fines. So below your bioretention media, if you have underdrains, you'll typically apply either a permeable liner. I don't like to use that. I prefer chokestone. We use a 4-inch layer and it works fantastic, but it needs to be free of fines. And so when they came out to construct the site, dumped the pea gravel in, and I know what it was spec'd to be. It wasn't free of fines. They had to dig it all back out. And the city didn't wait for me to say that. They were digging it out before I said anything. They knew we had a problem. Had to dig it out, had to go back and get it at the load. So making sure if you're going to use the chokestone layer and it's fantastic to use, make sure it's free of fines. The initial HPBSM rate is very high for infiltration. We were not expecting this, and which wouldn't have been a big deal in most cases. Who cares? It's going to go infiltrate faster, but our site is fully lined and all our effluent goes into that manhole. And then it goes through and has a pump that discharges it and pumps it to a dry well. The problem was we had a high intensity event coupled with a pump that was not sized for the infiltration, for the rate it was coming in at, that flooded our sump, destroyed pump. So we had a whole month of manual pumping and delays in getting started on our project. So I was in the middle of the night out there with my husband pumping. He married me better or worse. Part of that was pumping the site in the middle of the night, so we didn't mess up the site. So anyway, 650 DQ shipping delays. If you're going to the Manchester lab, it's considered a rural location. Who would have thought? So when you go to FedEx and they guarantee if you're there by 10, it's overnight, they'll have it the next morning. Baloney, that is not true. So because it's a rural location, it can take more than a day. So do not ship on a Thursday. Just wait till Monday and ship out your sample. And if it's late, your ice packed, container will still be good. We learned, and that was a tough one. The concrete edging. So we've got a non-vegetated site with basalt, a 3/4 inch chip. We went to the pre-construction meeting. Gonzaga is a partner. We're standing out there and Gonzaga's plant services says, I want a concrete berm around your site so that when we mow, we don't have rocks when we mow. Great idea. Would have been good to know when we developed the cost estimate for the project. So that was a last minute add-on. It looks beautiful. But if you do have a site where if you've got rock next to grass, you're worried about that, the concrete berm has been fantastic. So, and that is all. Thank you. No, so there's only one supplier of the HPBSM in Washington at the time we were speccing it out and getting ready, which is in Tacoma. So they had it all pre-mixed. Somebody from the city of Spokane drove over with a flatbed, picked them up and brought them back. So that was, we really looked, we had a list of items we could have used, but Gonzaga is very picky about how their campus looks. Like the green vault that was there, I'd picked out a red one, and plant services director was like, I do not want that. I want one that's green and blends in with campus. So I mean, they're kind of very specific about what they want with that. So we just went with that one. I think I would like a larger rock. And the reason I say that is because like with those seeds, how are we going to get those out of the rock? I'd rather have a larger rock that we might be able to use a blower that we could blow it out and then clean it up, because I think that's going to be a challenge with a small one. Oh, so it infiltrates, it comes in through one catch basin at the site. So it's, so I can go back to that. So this is the influent catch basin right here. And the entire 0.6 acres goes into that one point. It goes through where a pressure transducer is located and then a weir. So we get a depth that we can calculate the flow rate coming in, splits between the two cells. Then the two cells are fully lined with a permeable liner, infiltrates through the media, through the basalt into the media. Underneath we have the pea gravel and an underdrain, and an underdrain collects everything. And that discharges into the manhole itself. And then collect our information there and it's pumped out. There hasn't been a concern about it so far. It was a bioinfiltration pond.
Okay.
And then we split it. So we can get back to that earlier one. There we go. Yeah, it was this bioinfiltration pond. So you can see the two dry wells that were there.
Okay.
And then it still functions as that, but there's a full berm around our site. You can kind of see it in this middle picture here. Right here. You can see the berm. So we just get the runoff from the parking lot and the runoff that comes in from roof drains is outside that area and doesn't mix with it. It was over 35 inches per hour for that first one. I guess I wasn't expecting. I did contact Dylan. I'm like, what in the heck is going on here? It was insane to watch too, because the one pond is ponded and the other one is like, it's like quicksand. It was insane. And so we were on the phone trying to figure out what was going on. So yeah, anyway, Dylan. Not really. I was expecting to see that, because these are in order on that one, although I don't think I have the data. We'd have to go through it. We have seen in the past, again, that was one event. I guess I don't have all of I don't have all of those in here. For the metals, well, no, so it was event one, so the negative 168, it was a little bit higher. But for zinc, we're not seeing any big difference, and I know this is like 20 for hardness, so we're not seeing it there. I was thinking it was related to that. Again, that's one data point. We haven't always had the hardness because we weren't testing it in the past, so that'll be interesting to watch moving forward. No, we do have a rack set up in the front. I don't have it that helped to prevent that. We do sometimes have problems, but we haven't had, we go out and try to clean that out beforehand. That doesn't, I mean, in the fall, there's so many leaves, I can only clean so much, but we haven't had a problem with the bypass at all.